We are preparing for the SeekSpace single-cell spatial transcriptome seq experiment. Before starting the assay, make sure the Seq1 digital droplet system and all required accessories are properly set up. Power on the instrument and confirm that the system is ready for use. Next, review the subkits included in the SeekSpace single-cell spatial kit. The SeekSpace workflow uses multiple functional subkits. Let's take a closer look at each one. Spatial chip is used for sample slice attachment. Each chip has 2 labeling areas. Chambers allow labeling reaction and incubation. SeekSpace single-cell spatial labeling kit includes labeling reagents that should be stored at -20 degrees Celsius. The Chip S3 kit includes the droplet chip, gasket, carrier oil, and demulsification reagents. Store this kit at room temperature. Barcoded beads are used to capture and index RNA molecules. This kit must be stored at -80 degrees Celsius. These reverse transcription kits support both reverse transcription steps in the workflow, including the preliminary reverse transcription before droplet barcoding, and the second RT after decross-linking and cleanup. Store these kits at -20 degrees Celsius. Spatial barcode ligation kit is used for ligation reaction of spatial oligo. Store this kit at -20 degrees Celsius. Buffer S and Buffer T support nuclei processing and the preliminary reverse transcription workflow. DCL buffer and enzyme K1 are used during the decross-linking step after droplet recovery. Store this kit at room temperature. This kit provides the primers, indexes, and amplification reagents required for downstream cDNA and spatial barcode library preparation. Store these kits at -20 degrees Celsius. Cleanup beads are used in the purification step post-demulsification. Store this kit at 2 to 8 degrees Celsius. This kit provides 8 sample multiplex tags that allow one sample to be distributed across multiple tubes, supporting higher nuclei input and increased throughput. Store these kits at -20 degrees Celsius. TSO kit is used for template switching in pre-RT and secondary RT reaction. Store these kits at -80 degrees Celsius. In addition to the kit components, several user-supplied materials are required. Ensure access to standard laboratory instruments for sample processing, PCR, quantification, and quality control. A fluorescence microscope with slide scanning capability is required for spatial imaging workflow. The SeekSpace single-cell suspension preparation system is specifically designed for nuclei preparation in this workflow. RNA quality assessment is recommended before starting the workflow to ensure sample integrity. The reagents shown here support standard RNA extraction and quality control procedures, although alternative validated workflows may also be used. Prepare standard H and E staining reagents for tissue morphology assessment. High-quality sections should show intact tissue architecture without folding, ice crystal artifacts, or extensive necrosis. These reagents support nuclei isolation, staining, and sample handling throughout the workflow. Pay particular attention to RNase inhibitor usage. Approximately 40,000 units are required per chip. Single-stranded DNA dye is used for nuclei staining and downstream spatial alignment analysis. These reagents are used for library purification, fragment analysis, and sequencing quality control during downstream library preparation. Use certified low-binding consumables to minimize sample loss and contamination. Before proceeding with downstream experiments, confirm that the sample meets the recommended quality criteria. RNA integrity should meet the required threshold for transcriptome analysis. Tissue morphology should remain intact without major folding, ice crystal damage, or extensive necrosis. Finally, based on the H and E image, estimate the total nuclei count across the tissue section using image analysis software such as Cell Profiler. We recommend proceeding when the estimated count exceeds 100,000 nuclei per section. SeekSpace has been successfully validated across a wide range of tissues and species. Some tissue types may require additional optimization because of high water content, low RNA quality, difficult sectioning, or low cell recovery. The examples shown here are representative tissues only and do not constitute a complete compatibility list. For tissues not listed or for challenging sample types, please contact your local distributor or technical support team for evaluation and recommendations. Once all items on this checklist are confirmed, you are ready to begin the workflow.